So now that we have retrieved our FASTQ file, now we are done with the first part, which is the raw reads, and now we are going to move on to the quality control. Quality control of our raw reads. Now that you know that NGS data came from several other sequencing platforms like Illumina, Biopack, and others sequencing companies like that. So PacBio is one of them. So we are going to check if our data is correct or not. Our data contains that quality to process further for the data analysis or do not process further for the data analysis. So moving on, what quality control basically is. So first we're going to learn what FASTQ file is, because in the last lecture we left it here. So here you can see that the FASTQ file contains reads like this. We can also visualize it on our file. We can get the file.fastq. Here you can see there is a session number, there is length, there is sequences. Also there is length mentioned here. So moving on to the here, you can also see that there are different other flow cell ID which was used and sequencing, which lane we were using, which tile it came from, which coordinate it contained, what are the read bases, what are the read bases scores. Also there is separator that is optional repeated header. Also we contain these kind of files in our FASTQ file. So that was it for the FASTQ file format. So what is quality control? So and which tool gives us the FastQC main description. So FastQC provide a simple way to do some quality control checks on raw sequencing data coming from high throughput sequencing pipelines, high throughput sequencing companies. It provides us a set of modular analysis which give a quick impression of whether your data has any problem of which you should be aware before doing the further analysis. For example, if our data contains repeaters, or if our data contains a lot of sequencing different variants that we gonna check if they are okay to deal with or if they are not. If there are so many adapters that are added to our sequencing data. So these kind of stuff we're gonna check with the FastQC. So what are the main functions of FastQC that we use? So we're gonna import our data into SAM, BAM, or FASTQ file, any variant we can use. So we will be checking we will be getting a quick overview of which areas have problem. So summaries will be created like the graphs and table for our data, and then we can export the results. So how to do this kind of quality checking? For example, we have the FastQ file. We're gonna check. We're gonna install the FastQC tool. We have FastQ file. We're gonna check the quality control. We're gonna do some quality control in FastQ file. For that, we need a tool which is FastQC. So FastQC is a simple way to do so quality checking. So we're gonna install our tool, which is the FastQC tool, and then we're gonna quality control some data of FastQC. So these are the two commands that we are going to use for installing and updating our system. Moving on to our text file, I'm gonna paste it here. Sudo apt get update, I'm gonna update my system, and then I'm gonna install the sudo apt get install FastQC. So that is it for installing our FastQC tool. Now that FastQC tool has been installed, we need some file for the data analysis. For example, we need the FastQC file.fastqc, which is already we have our file.fastqc. FastQC is a tool that we need. FastQC. I'm gonna check. Yep, it is installed. FastQC tool is installed. It requires some Java files that it is seeing. And for checking the quality, now that we are in this folder, we have the file.fastq. We're gonna check our data quality. We're gonna make a directory mkdir, which I'm gonna name data, and now I'm gonna check. Yep, data folder is created. I'm gonna apply the FastQC tool, and then I'm gonna write the write a quick overview of that, and I'm gonna check our file.fastqc, then gonna generate a output into data folder, data and /. So it is saying that analysis complete for the file.fastqc. So it is saying the analysis completed for the file QC. I think I have done something wrong. I should have included the data. Yep, sorry, I should have included the O. I included 0 there. So that was the main problem that we're causing. So here we got the FastQC quality control in our folder. Now I'm gonna ls, and I'm gonna move to the data folder. Moving data, and I'm gonna check here. Yep, file.fastqc HTML are the results that we need. So move into this folder, we have our data. Here you can see file.fastqc. So the file name that we used was file.fastqc. It has conventional base cell, so encoding was done by Sanger sequencing. Illumina platform was used 1.9. Total number of sequences that we cover were 2, and the sequence length was 72 base pair. Because it was a demo testing, it was a demo sequences, there were only 2 sequences there, so GC content was not enough. Our basic stat was enough. Our per base sequence quality was okay, and our per base sequence score were not okay because we contained so much data, so less data. So per base sequence content was not enough, n base content, GC content was not enough, and overrepresented sequencing, there were no hits as well. And adapters, there were also no adapters found, so we don't need trimming, but we are going to do that trimming as well, because we gonna use this pipeline in your data. So you need to have, or you need to know how to apply trimming process on your FastQC file. So that was it for the FastQC report. Now you know how to check the quality control of your data, and that was it for this lecture as well.